Hello friends and welcome back to my channel Micro and Mall Bio with Dr. Sunita Bundale. In one of the previous lectures, we have already talked about the pioneers of microbiology, particularly the contributions of Louis Pasteur, Robert Koch and Edward Jenner in the field of microbiology. But I believe that the science and the field of microbiology is incomplete without the discussion of some other important scientists like Joseph Lister, Elie Metchnikoff, and Paul Ehrlich. From sterile surgery to the birth of immunology and chemotherapy, these visionaries transformed the way we fight diseases. So let us talk about these great scientists in this particular lecture today.
Joseph Lister was a professor of surgery at Glasgow University. He was worried about the fact that many people survived the trauma of an operation but died afterwards due to post-surgical infections known as ward fever. In the Edinburgh hospital where Lister worked, almost half of the surgery patients died from such infections. Lister had read about Pasteur's work on how wine went bad because of microorganisms in the air. Lister wondered if the microorganisms in the air were also the cause of the infections which killed his patients after they had successfully survived surgery. Probably the open wounds made it easy for germs to get into the body. He started to clean the wounds of his patients with carbolic acid and soak the dressings in the antiseptic liquid as well. He invented a machine that misted out portions of carbolic acid into air around the operation table and equipment to kill airborne germs. He then experimented with hand washing, sterilizing instruments, and spraying carbolic acid in the theatre while operating in order to limit the infection. Note the person spraying carbolic acid in the operation theatre during surgery in the photograph. In the years from 1864 to 66, the death rate for Lister's surgical patients was 45%. However, between 1867 to 70, when he introduced his new antiseptic treatment, this fell drastically to 15%. Lister was very eager to acknowledge Louis Pasteur's invaluable contribution. In a letter to Pasteur in February 1874, Lister wrote, "Thanks for having, by your brilliant researches, proved to me the truth of germ theory. You furnished me with the principle upon which alone the antiseptic system can be carried out." Today, although the antiseptics and disinfectants being used have changed, aseptic surgery is still the basis of saving millions of lives, and Joseph Lister is rightly called the father of antiseptic surgery. Elie Metchnikoff was a Russian who was one of the first scientists to study the body's defense against invading microorganisms. He served as a professor of zoology and comparative anatomy. It was in Messina, Italy, while studying the digestive organs of starfish larvae that he discovered phagocytosis. One morning in 1882, as Metchnikoff observed cells moving freely within transparent starfish larvae, he hit upon this theory. These wandering cells in the body of starfish larvae, these cells eat food, but they must also eat up microbes. Of course, the wandering cells are what protect the starfish from microbes. Our wandering cells, the white blood cells, must be what protects us from germs. To test this hypothesis, he inserted small thorns from the garden plants into the starfish larva. The following morning, he noted that the cells within the larvae were no longer moving around aimlessly but were instead aggregated around the foreign bodies as if to drive them out. The theory of phagocytosis was thus taking shape. He theorized that under appropriate conditions, protected cells within an organism would mount an immune response by attacking and ingesting foreign matter. Metchnikoff coined the term phagocyte from the Greek words phagos meaning to eat and cyte meaning cell. But phagocytosis, the cellular basis of immunity, took a temporary backseat when German scientist Emil von Behring discovered humoral immunity in 1890. An intense debate erupted as to which theory was correct, with the French supporting Metchnikoff and the Germans von Behring. Eventually, both theories became entrenched as being fundamental to the study of immunology. Metchnikoff then moved to France in 1882, adopted it as his home, and chose to work at the Pasteur Institute in Paris. During his last 15 years, Metchnikoff became fascinated with the biology of aging and death. He believed that the aging body was gradually poisoned by waste products from harmful bacteria in the intestines, which could be neutralized by lactobacilli in yogurt and sour milk, and strongly advocated regular ingestion of lactobacilli. Thus, he was pivotal in starting the relatively modern discipline of probiotics. Metchnikoff received the Nobel Prize in Medicine in the year 1908 for his work on cellular immunity along with Paul Ehrlich. Talking about Paul Ehrlich, he was a German medical scientist known for his pioneering work in hematology, immunology, and chemotherapy, and for his discovery of the first effective treatment for syphilis. He was obsessed and fascinated with the affinity of stains and dyes for tissues and the underlying chemistry of the reaction that he pursued his doctoral research on contributions to the theory and practice of histological staining. When Koch had announced the discovery of the tubercle bacillus in 1882, Ehrlich lost no time in developing an effective strain to demonstrate the tubercle bacillus and establish its acid fastness. Ehrlich continued to experiment with cellular staining. Ehrlich showed that all the dyes used could be classified as being basic, acid, or neutral, and also differentiated the numerous types of blood cells of the body. His work on staining of granules in blood cells laid the foundations for the field of hematology. The selective actions of these dyes on different types of cells suggested to Ehrlich that chemical reactions were occurring in cells and that these reactions formed the basis of cellular processes. From this idea, he reasoned that chemical agents could be used to heal diseased cells or to destroy infectious agents. He prophesied that chemists in their laboratories would soon be able to produce substances that would primarily seek out specific disease causing agents. He called these substances magic bullets. Little did he realize that it would be a 17-year-long journey before he would develop salvarsan, the first effective chemotherapeutic agent against Treponema pallidum, the causative agent of syphilis. Salvarsan was the 606th chemical agent he tried and was extraordinarily effective and harmless despite its large arsenic content. It seemed as if the magic bullet had been found. Ehrlich did further important work on immunology and showed that the toxin antitoxin reaction is accelerated by heat and retarded by cold, and that the content of antitoxin in the antitoxic sera varied so much that it was necessary to establish a standard by which their antitoxin content could be exactly measured. This work and his other immunological studies led Ehrlich to formulate his famous side chain theory of immunity. He received, jointly with Elie Metchnikoff, the Nobel Prize for Physiology and Medicine in 1908 for the discovery of salvarsan.